professor here at our Lombardi Cancer Center and a clinical investigator with a long-standing track record in funded breast cancer research. She worked for over a decade as a breast oncologist and investigator at the University of Toronto in Canada. She then moved to sunnier climes at the University of Miami to the Sylvester Comprehensive Cancer Center, where she founded and directed the Bremen Family Breast Cancer Research Institute. She also served there at the Sylvester Comprehensive Cancer Center as the Associate Director of Translational Research and Breast Program Leader. In 2020, she moved to Georgetown and serves as the co-leader of our breast cancer research program and is a professor in oncology. She discovered the cell cycle inhibitor P27 and has contributed to the understanding of G1 to S phase transition and how aberrant signaling via TGF-beta, SRC, MAC, and P13K pathways disrupt cell cycle regulators in cancer cells. She studied the use of targeted therapies to reverse anti-estrogen resistance in breast and ovarian cancer and has brought these to clinical trials. She's worked in and focused on the understanding of how hierarchies in breast cancer cells contribute to chemoresistance and may be targeted by novel epigenetic drugs. So with that, I look very much forward to hearing about her work.
So it's my pleasure to give grand rounds this morning. I was recruited in the midst of COVID, so haven't had the opportunity to meet with many people in person. Can you hear me okay?
Yes, we can.
Yes,
Good.
perfectly. And the slides look great.
Good, and you can see my pointer?
Yes.
Good, okay, great. So thanks for the opportunity to speak to you, and I'm going to share an area of research that really we've looked into over the last 6 or so years, and that really is how does obesity impact breast cancer risk and mortality. Obesity has become sort of the recent health scourge of the world, indeed. In the USA in 2017, approximately 39% of adults were obese, and it's been estimated, very informative paper from Ward's group in 2019 indicated that by 2030, there would be at least half of the American population with a BMI in the obesity range and potentially a quarter of the population morbidly obese. Currently, approximately 70% of all US women and 40% are overweight. And overweight in breast cancer patients has been somewhat hard to estimate. Lidwell published an estimate of over 74% of breast cancer patients were overweight in 2014, so the number is certainly higher. So obesity is categorized as a body mass index of greater than 30 kilograms per meter squared, and over 35 is considered morbidly obese. Worldwide obesity has tripled since 1975. In 2016, nearly 2 billion adults were overweight, and of these, 650 million were obese. And there's a rising incidence of obesity in children, and this is a harbinger of health problems to come. And it would be fair to say that most of the world's population live in countries where overweight and obesity kills more people than underweight. And this is just a 2014 map showing a very significant proportion of the globe has significant numbers of adults in the obesity range of BMI. This is the map of the United States of obesity, and as we all know, blue is good and red is bad on the map. Especially from recent events. But 1990 had a the obesity rates in the States rarely exceeded 10 to 15%. By 2010, approximately 1/3 of the nation had a BMI greater than 30. Now the dark red is still problematic on the next map, but it represents greater than 35%, and this color is now greater than 30. This is the profile in 2019 for non-Hispanic whites in the nation. And you can see that that corresponds to the graph I showed you on the previous slide. And you can see that there's been a considerable expansion in the population with a BMI greater than 30. So in a considerable portion of the states, greater than 35% of the population has a BMI over 30. And, there's been a very significant observation that non-Hispanic Blacks have a greater incidence and mortality of breast cancer. And I'd like to point out that there's a greater incidence of obesity also within this, within non-Hispanic Blacks in the nation. This is the graph for children showing adults have gone up in their BMI between 2000 and 2015, from approximately 30% to 39%. But look at the youths. So between the ages of 2 and 19, approximately 14% were obese in 2000. That's gone up to nearly 20%. And there's a disparity among children in obesity, with a disproportionate number of Hispanic children and Black children affected by obesity. So this may be a harbinger of a future increase in cancer burden as we go along if we're not able to reverse this pattern. As long ago as approximately 20 years ago, it was appreciated that cancer patients who are obese have higher mortality. And this is a landmark paper by Elizabeth Cowley in 2003. And for breast cancer, there was an increased reported rate of mortality of 2.2. It's been thought that up to 20% of all cancers may be attributable to obesity, and 50% of deaths from breast cancer may be attributable to obesity, and it could be higher. Obesity associates predominantly with a higher risk of ER-positive breast cancer after menopause. There's an increased risk of breast cancer in individuals with metabolic syndrome, and for all types of breast cancer overall, after menopause, there's an increased risk. Meta-analysis of over 2.5 million women showed a modest increase in relative risk with elevated BMI. For inflammatory breast cancer, which is a relatively rare but very aggressive subtype of breast cancer, there's a nearly fourfold increased risk. But the most important increased risk associated with obesity is for postmenopausal women with regard to development of estrogen-responsive ER-positive breast cancer. And notably, this type of breast cancer is the most common breast cancer. And indeed, it's this type of breast cancer that leads to the greatest numbers of patient death, even though in itself, it's not the most aggressive form of breast cancer. And this is just a review article we made showing a forest plot of multiple different prospective cohort studies that were reasonably well powered, showing relative risk of development of breast cancer, of ER-positive breast cancer, ranging between about 1.3 and 2.7, and the overall meta-analysis that showed an overall risk, relative risk of 1.4. So breast cancer specific, so in addition to increasing the risk of developing breast cancer, obesity is also associated with a higher breast cancer mortality regardless of the subtype of breast cancer that there is. It's approximately twofold increased risk if the BMI is in the morbid obesity range. High BMI in breast cancer studies correlates with larger tumors, has been strongly associated with greater numbers of NOBs positive, and an increase in a more aggressive form of breast cancer, the triple negative, lacking estrogen receptor, progesterone receptor, and HER2. These patients have more complications of surgery, chemotherapy, and endocrine therapy, and frequently it's been the worst outcome has been attributed to undertreatment, particularly in the area of chemotherapy administration, since we tend to cap our levels of chemotherapy calculations that are calculated by BMI at 2 meters squared, and obese women often have a much higher body surface area. So recent meta-analysis involving a very significant number of studies in patients showed breast cancer death is also upregulated by about 1.4-fold in obese women. And this is again a forest plot from that recent meta-analysis showing significant, essentially all studies showing higher risk of mortality. So we were really interested in what is it that's bringing about, what's the underlying cause of this? There'd been a lot of noise in recent years about how when breast cancer is invaded into surrounding tissue, the fibroblasts and the stroma influence the development or the progression of the malignant cells. But if you look at a breast cancer morphologically, this is what we often see. We see the tumor cells surrounded by mature fat cells, and there's a direct interface between the cancer cells and the fat cells. And we wondered what happens, and can we mimic this in the lab and see what's going on? As we got into this work, we realized that obesity leads to a chronic inflammatory state in multiple tissues, but particularly in obese adipose tissues. And this can be seen by marker crown-like structures. So these crown-like structures represent dying adipocytes that are undergoing lipolysis, and they're surrounded by infiltrating macrophages. These crown-like structures increase with age, they increase with obesity in breast fat, and they also, in the mammary gland, the evidence of these crown-like structures in mammary fat correlate with poor breast cancer prognosis. So what's going on with this inflammation, and how is it linked to our increased risk and poor outcome? Well, it turns out that obese adipose tissue is a pro-oncogenic environment. With increased proliferation of adipocytes and decreased vascularization, there's hypoxia, which induces hypoxia-inducible factor alpha, which is a transcription factor. This transcription factor leads to downregulation of adiponectin, upregulation of the mitogenic leptin, and upregulates also secretion of cytokines that themselves act as mitogens and vasculogens in developing fat. So there's a shift towards an abundance of pre-adipocytes that are little cytokine tanks in obese fat. And lipolysis further stimulates the NF-kappa B pathway via TLRs, leading NF-kappa B is a critical pro-inflammatory pathway that's characterized by specific transcription factors that often drive particularly the expression of these cytokines. And this milieu then leads to attraction of monocytes that are converted to macrophages, particularly in the high leptin environment, shifting the profile of immune cells in obese fat towards a pro-inflammatory environment, and interestingly, leading to a downregulation of immune mediators of anti-tumor immunity. So that's sort of the headlines, and now I'm going to tell you the news. So this is just a picture that paints the same thing I just told you on the last slide. With decreasing oxygen content, HIF-1 alpha goes up, shifts to upregulate leptin and repress adiponectin, turning on VEGF that drives vasculogenesis. And as lipolysis takes place around those crown-like structures, the free fatty acids stimulate toll-like receptors, driving NF-kappa B, upregulating these cytokines that are mediators of inflammation. And NF-kappa B also turns on aromatase, an enzyme that will be part of the story as we go along, which leads to an upregulation of available estrogens in fat. So our story in my lab started about 6 years ago when we looked at the interaction of adipocytes with cancer cells. And this summarizes quite a significant amount of work, but what we found is that the adipocyte and the breast cancer cell undergo a pro-inflammatory reaction when they bump into each other. And these experiments were done by plating immature adipocytes right on top of cancer cells in culture in the lab. And what we found is that activated the SRC-ONC gene, which is a signaling molecule leading to upregulation of these pro-inflammatory cytokine gene expression. And the cytokines themselves, in turn, were needed to keep SRC activated. So it led to a feed-forward activating loop that turned on a number of genes that are critical regulators of stem cells. And these stem cells are very oncogenic in breast cancer models, leading to an enrichment in the most aggressive kind of breast cancer cell called the cancer stem cells. So cancer stem cells were being upregulated by a feed-forward loop that occurred as a result of interaction between fat cells and cancer stem cells. So we wondered, how do estrogens affect this story? Because estrogens are really important mediators in adipose tissue, particularly in the context of obesity. So if you'll recall, after menopause, our main source, ovarian estradiol, is decreased, and our main source of estrogen after menopause for women is the conversion of adrenal androstenedione, which is synthesized at a pretty constant rate throughout life, not much affected by age or obesity. But the androstenedione gets converted to estrone via aromatase, and that takes place in the bone and in the brain, in the breast, and predominantly in fat. So aromatase is highly expressed in adipocytes, and obese women have up to 3-fold higher circulating estrone in serum and even higher levels in breasts. So in breast tissue, the elevation of estrone may be even higher than 3-fold with obesity. There is decreased sex-binding globulins, so estrogens are more bioavailable, and it's been shown that obese women with the highest quintiles of serum estrogens have considerably higher, that estrogen accounts for their higher risk of breast cancer. The NF-kappa B inflammatory pathway that I've told you is activated in fat also turns on aromatase, and some of the products of NF-kappa B activation, TNF-alpha, also stimulate aromatase activity. So we wondered, maybe this very high estrone is a driver of the aggressive behavior of the adipose environment and its effects on breast cancer cells. So what we did is we took reduction mammoplasty specimens of fat. So we've got fat from the OR, from plastic surgery waste material, and we compared non-obese premenopausal breast fat with obese breast fat. And what we found is that there was a significant increase in a number of pro-inflammatory mediators with obesity in individuals who were before menopause. We also looked at in women undergoing bilateral mastectomy for breast cancer, we recovered breast fat from the contralateral unaffected breast and the breast cancer affected breast. And what we found is that peritumoral fat compared to the contralateral normal have elevated expressions of these pro-inflammatory cytokines. And when we compared pre and postmenopausal fat, both from obese donors, the postmenopausal fat was more inflamed. So there seems to be potentially an interaction with hormonal regulators in driving the inflammation. And when we mixed our premenopausal versus postmenopausal fats with cancer cells, so the cancer cells, MCF7 estrogen receptor positive cancer cells, have very little expression of these cytokines on their own, but when they come in contact with adipocytes, the adipocytes from the premenopausal women, yes, indeed, led to a significant upregulation of these inflammatory mediators, but the postmenopausal breast fat contact even further stimulated the expression of these inflammatory cytokines. And so what's the difference between premenopausal fat and postmenopausal fat in terms of estrogen? Well, the postmenopausal fat has a way lower estradiol. Interestingly, breast fat, the breast appears to guard, even despite the loss of estradiol, breast fat guards its levels of estradiol, and there's similar levels of both estradiol and estrone. E1 is the abbreviation for estrone, E2 is the abbreviation for estradiol. So we next look to see whether indeed this upregulation of cytokines was dependent on estrogen. So here what we've taken is we've taken our cancer cells and we've taken our cancer cells that were co-cultured with the adipocytes. Those are the white bars. So the cancer cells had very little cytokine production. When we co-culture them with the fat, the cytokines go up. And if we pre-treat those with letrozole, which is an inhibitor of aromatase and prevents estrogen from being synthesized, we saw a much lower level of cytokine induction. And similarly, if we pre-treated our adipocytes and cancer cell mixtures with NF-kappa B inhibitors, we found that the upregulation of the cytokine on co-culture was considerably less in the presence of the NF-kappa B inhibitor. So this induction of cytokines is a cooperation between estrone and NF-kappa B driven pathways. And the reason I say estrone rather than both estrogens E1 and E2 is that we actually measured what's left over within 48 hours and 6 days of co-culture. And very shortly, you realize that the co-culture media is really dominated by the presence of estrone. So how does this interaction between estrogens and the NF-kappa B pathway take place? So estrogen binds to the estrogen receptor, and in the context of obesity, the abundance of free fatty acids produced by excess lipolysis are continuously stimulating the TLR4 receptor, which is leading to dissociation of this inhibitor from the P65 transcription factor, allowing it to whip into the nucleus and turn on genes. And it's been known for some time that estradiol actually attenuates inflammation in many human diseases such as MS, inflammatory bowel disease, rheumatoid arthritis. Estradiol is anti-inflammatory, and we know that it's also neuroprotective. It inhibits NF-kappa B luciferase. That's the NF-kappa B promoter sitting in front of a luciferase gene, and you can measure induction of that promoter in vitro in multiple cell types. And it's known that estradiol binding to the estrogen receptor actually inhibits NF-kappa B's ability to turn on the CCL2 promoter. And what happens is that the estradiol-bound estrogen receptor either kicks off the P65 molecule from the promoter of the CCL2 gene, or recruits co-repressors, or fails to recruit co-activators. So nobody had really looked at how this interaction takes place with estrone. The only estrogen that's really ever been studied in these regards is estradiol, the premenopausal, most abundant estrogen. So what had been shown is that whereas normally estrogen interacting with the estrogen receptor binds to estrogen response elements, there's a special kind of gene response to estrogen that takes place in the context of inflammation. So if you mimic inflammation by flooding the cells with TNF-alpha, the estrogen receptor gets recruited together with P65 to specific pro-inflammatory genes. So in the context of inflammation, the estrogen receptor shifts away from estrogen response elements and onto kappa B sites. So we're very interested in knowing how does estrone affect this. And that's a little bit of the story I'm going to tell you. So there are only about 5 more data slides, and then I'll get on to clinical application. So we then looked at how does estrone differ from estradiol in how it turns on genes. So we did RNA sequencing experiments using either estrogen-deprived cells, to which we then added either estrone or estradiol. And this shows you the Venn diagrams of the genes that were upregulated or downregulated. And you can see that most of them are the same. There were about 12% of genes uniquely upregulated by estrone, and about 22% uniquely downregulated by estrone, and similarly small proportions uniquely up or downregulated by estradiol. But even in the genes commonly up or downregulated, the patterns of individual genes, this is the extent of upregulation shown as red, blue is down, and you can see that the extent of upregulation across individual genes that are displayed horizontal here is very different. And similarly, genes that are downregulated, if you look at the profile for individual genes across the horizontal, there's a real difference in the pattern. And one of the things that was uniquely upregulated by estrone in this gene profiling analysis by RNA sequencing was the TNF-alpha pathway was uniquely activated by estrone, but not by estradiol. And to understand how does this happen, we looked at individual pro-inflammatory cytokines. So the control cells don't really turn on. This is the RNA expression. E1 and E2 on their own don't really turn on these pro-inflammatory mediators in breast cancers. In our breast cancer model we were working with, and we used 3 different models. So TNF-alpha is the immune activator that turns on the NF-kappa B pathway, and CCL2 gets activated. But when we added estrone, we saw a much bigger activation of that gene. It was blocked by fulvestrant, showing that it's carried out by estrogen binding to the ER, the estrogen receptor. When we added E2, the estradiol, the premenopausal estrogen, lo and behold, we didn't get a stimulation of TNF-alpha mediated CCL2 induction, but rather it was repressed. How was that happening? So then we looked at how do the transcription factors get recruited to the promoter of the CCL2 gene. So that was done by a CHIP assay. And you can see that there's no, so this is the P65 NF-kappa B transcription factor. This is the estrogen receptor alpha, and this is a co-activator necessary for transcription. So in the control cells, we didn't get much activation. When we added the pro-inflammatory mediator, both P65 and ER are recruited to that promoter site. When we add estrone, the recruitment is modestly upregulated for P65 and not much different for ER. But when we look at what's happening to the coactivator, this is an essential protein necessary for that complex to bring together to get the gene turned on. And what we found is that the estrone is bringing in the coactivators, but the estradiol is booting them off that promoter. So estrone is cooperating with the NF-kappa B pathway to turn on genes. And we're very interested. We've begun to do global ChIP-seq RNA sequencing experiments to look at the global patterns of how these two transcription factors are recruited to promoters and what are the differences in how they turn on genes. And CCL2 is an important inflammatory mediator. There are a host of these genes, and we believe that this pattern is likely going to play out with a whole host of pro-inflammatory mediators as a way that estrone is really, after menopause, when we lose estradiol, there's a big difference in how we respond to inflammation. And that may be a factor leading to greater risk for development of breast cancer. So more adventures of that come from looking at genes and how they differ. So this is again from the RNA sequencing showing a pattern of genes that are upregulated by TNF-alpha or the inflammatory mediator. When you add estrone, a whole profile of genes is even more greatly upregulated. And these same genes, when you add E2, don't get turned on. So we're very interested in this kind of gene to see whether they're mediators of bad activity in breast cancer. So we did gene ontogeny, looking at what patterns of genes are upregulated by these particular genes. And they're involved in signaling through mTOR, insulin resistance, cytokine signaling, pathways relevant to cancer, and the like. And indeed, we could discern from among these genes a profile of genes whose upregulation, these are the T plus E1 activated genes, their expression at high levels in breast cancer patients correlated with worse outcome, not only in the Metabric, but also in the TCGA data sets. So there's something about these inflammation plus estrone-driven genes that makes them potentially risk factors or mediators of poor outcome in breast cancer. So the next thing we did is to try to garner evidence that there may be a difference in what E1 and E2 do in vivo in mice. So here we used transgenic mice in which the NF-kappa B promoter is transgenically expressed in all cells of the mouse's body, driving the luciferase promoter. So when NF-kappa B is activated, these little mice glow green. So here we put our control mice on a low-fat diet and our test mice were on a high-fat diet, and indeed they gained weight. And what you see here is the in vivo imaging system that the little green mice are the ones that are inflamed. And what you see here is that this is just showing the photon flux, which is a measure of the luminescence in these mice. And you can see that the animals on the high-fat diet had greater levels of inflammation compared to the low-fat diet. But the animals that were on the high-fat diet and stimulated with estrone had way higher levels of inflammation as measured by NF-kappa B activity. And you can see that the high-fat diet estrone-treated mice glow green compared to the animals that were on that were either the control or treated with estradiol. So estradiol, in contrast to estrone, that augmented obesity-related inflammation, estradiol did the opposite. So that was a fairly nice in vivo evidence that there is a difference in how these estrogens interact with inflammation, with estrone, the postmenopausal estrogen, being very pro-inflammatory compared to what we have before menopause. So recall that I told you androstenedione gets converted to estrone through aromatase. Well, there's a fairly significant exchange of estrogens and indeed androgens in the body regulated by the hydroxysteroid dehydrogenase enzymes. There are 14 of these enzymes responsible for interconversion of one type of estrogen to another. And interestingly, their biological distribution in the human body is very poorly understood. And what regulates them is even less well understood. But they're really critical for understanding local steroid levels in biologically important tissues like the breast and the fat. And what we posited that if estrone is pro-inflammatory and is a mediator potentially of inflammation-driven tumor progression, it's possible that breast cancers that are hormone sensitive, if they had higher levels of the enzymes that drive estrone upregulation, might do worse. So if E1 is the bad actor, an enzyme that drives increased E1 might correlate with worse outcome. So we looked into these large databases of breast cancer, and you can look at any one enzyme and see if it correlates with bad outcome. So in estrogen receptor positive breast cancer, this is a Kaplan-Meier survival curve. And what we found indeed that high levels of expression of this estrone driving enzyme correlate with about a 1.6-fold increased risk of death. So that was compatible with our hypothesis. So to investigate this experimentally, we introduced into 3 different estrogen receptor positive breast cancer lines the gene encoding this enzyme. So here you can see that they express the enzyme nicely, and when we measure the estrogen levels in the cells, the cells that are expressing, and I'm going to abbreviate it to HSD, even though that there are all sorts of them, but here I mean HSD17B14. And so they have lower levels of estradiol and higher levels of estrone. What does that do to their behavior in terms of cancer-related assays? Well, what we found is that just merely overexpressing this estrone-producing enzyme led to an upregulation of embryonic stem cell transcription factors that are known drivers of cancer stem cells, the more aggressive form of the subset of breast cancer cells that are very aggressive and drive metastasis. They also led to a generation of greater numbers of tumor spheres, which is also an indicator of cancer stem cell behavior, and upregulation of the aldehyde dehydrogenase enzyme, which is also a marker for that cancer stem cell aggressive population. And we directly tested E1 and E2 and found that indeed E1 upregulates and E2 downregulates these spheres. So it looks like, and in addition, when we compare gene expression profiles on RNA sequencing between the HSD overexpressing cells compared to the parental cells, a host of cancer-related bad signaling pathways and stem cell signatures were upregulated. And then the proof of the pudding is, how do these estrogens compare when we look at tumor growth in vivo? So here we grew cancer cells in vivo, and we compared the parental estrogen receptor positive MCF7 cells. These are the cell line that Mark Lippmann worked with in the 70s, and it was Mark's idea to say, Joyce, you really just need to get on with testing estrone versus estradiol in vivo. So we did that. And we also compared, so we had the MCF7 parental cell line, and here's its growth with estradiol. It does indeed stimulate growth. Here's the growth with estrone, same amount of estrone available, and they grow faster. And these 2 variants are the ones overexpressing the enzyme that takes whatever E2 is around and drives it into E1. And these grew like weeds. We also tested in another model, the EO771 model, which is a syngeneic breast tumor model, and we found same finding. E1 drives faster tumor growth than E2. and their actions were all blocked by fulvestrant, showing that this is working through action of the estrogen receptor. And it's interesting that there is interconversion of the estradiol and estrone within these tumors. And you can see that we measured intratumoral estrogen levels. So this is the estrone level, this is the estradiol level. The estradiol-driven tumors obviously had the highest levels of estradiol, but even the estrone-driven tumors were converting some of that estrone into estradiol. In contrast, and the estrone-driven tumors had high levels of estrone, but the estradiol-driven tumors also were able to take some of that estrone and convert it back to estradiol. So it's looking like the slowest growing tumors and just the HSD overexpressors had very little estradiol at all and high levels of estrone. So it would seem that the ratio of E1 and E2 are important. Having high E1 as a driver, but also attenuating your E2 seems to be propitious for cancer growth. It's even possible that, although this might be heretical to say so, it's even possible that the growth stimulated by estradiol results from its intracellular conversion into estrone. And we're trying to interrogate this experimentally as we go along. We wanted to enumerate the cancer stem cells in the populations of these different cells in the presence of estrone or estradiol. And to do that, what you do is you make limiting dilutions of the cancer cells, inoculate them into 10 mice each, and then look at how many of these animals actually develop tumors over time. And you can see this pink line here in both the 10,000 and 20,000 cell groups. The pink cells were the slowest to develop tumors over time, and also fewer of the animals actually develop tumors over time compared to so these are the estradiol stimulated tumors, and these are the estrone stimulated tumors. And whereas in the presence of estradiol, only 1 in 70,000 cells is capable of generating a tumor, in these estrone-stimulated tumors, as few as 4 to 18,000 cells could generate, 1 in 4 to 18,000 was capable of generating a tumor. And the tumors that emerged after estrone stimulation compared to estradiol stimulation were much enriched for this stem cell marker, or the cancer stem cell population. So to summarize, obese breast fat is inflamed, more so after menopause. We showed that in our reduction mammoplasty specimens. Invasion into adipose tissue upregulates estrone and NF-kappa B-driven cytokines. And we showed that in the obese mice, estrone activates and estradiol decreased the inflammation associated with obesity and diet-induced obesity. We found that estrone, but not estradiol, activates the NF-kappa B pathway on our RNA sequencing. And we found a mechanism that the estrone is bringing in a whole lot more of that coactivator to the complex of the estrogen receptor and the kappa B at the promoter sites that drive these pro-inflammatory cytokines. So, and finally, we showed that in regard to ER-positive tumor growth, E1 appears to give greater bang for its buck per similar concentrations than E2. And it appears that the ratio of E1 to E2 may be very important, augmenting tumor growth, tumor inflammation, I didn't show you that data, and enrichment for cancer stem cells. So the cancer stem cells are the ones that seed metastasis, resist chemo and radiation, and are really, we believe, the ones that we need to find better ways of targeting in order to improve patient outcomes. So we've found that postmenopausal estrogen appears to be a significant driver of the development and progression of cancers through these mechanisms. So what do we do from here? Clinical implications are that ER-positive breast cancer risk, interestingly, it's been known that in obese premenopausal women, as opposed to postmenopausal women, obesity seems a little bit protective or is associated with a lower risk of ER-positive breast cancer. And it may be that before menopause, the estradiol opposes the inflammation of obesity, and that anti-inflammatory effect of estradiol is lost after menopause. The other implications for this work clinically are we need to rethink estrogen replacement therapy. It's possible that if estradiol really has beneficial effects to protect against heart disease, to protect against bone demineralization, to have neurocognitive protective effects, if we could prevent its conversion to estrone, we might actually change endocrine replacement therapy to be predominantly estradiol with drug mediators that block those HSD17, B14, and 10 enzymes. And there may be a way of targeting estrone if we could develop ways that specifically interdict either production or the effects of estrone. We may have beneficial effects for diabetes, cardiac disease, and neurodegenerative disorders. There's a real need to understand how these 17 HSD beta enzymes are expressed and regulated. We really have very little knowledge of this. And then finally, as obvious as the nose on your face, is the need for lifestyle interventions for overweight and obese breast cancer patients. There are studies ongoing. There are a number, at least 5 very large multi-institutional international trials that are ongoing to test whether lifestyle interventions impact outcome for breast cancer patients. I wanted to spend a moment before leaving on impact of exercise and lifestyle intervention in breast cancer patients and tell you a little bit about where our work is leading to us in future. So it's known that a majority, wide variability in what proportion of breast cancer patients gain weight after diagnosis. And there's very nice epidemiologic data that shows that weight gain in breast cancer patients, which is so viciously common, is a factor in increased death risk from breast cancer. So each increment of 5 pounds is associated with a 12% increased risk of death for breast cancer from epidemiologic studies. And indeed, I mentioned before that obesity is also, and weight gain is associated with greater incidence and severity of treatment complications. The Women's Health Initiative found that women who breast cancer risk is decreased in the women who have the highest levels of physical activity. So physical activity is associated with reduced risk of developing breast cancer, in addition to, and meta-analyses have shown that the highest pre-diagnosis physical activity is associated with lower HR for death. And these are very big meta-analyses, and this is true for both breast as well as colon cancer. And all patients benefit regardless of any subgroup. Another really interesting study was one from the Women's Health Study that showed that 3 to 5 hours of moderate physical activity, such as a brisk walk, are actually in women after diagnosis of breast cancer. So those women who were able to maintain or who had adopted a pattern of moderate physical activity had up to 50% decrease in risk of death. And that translated to an absolute reduction in mortality at 5 years of 4%, very similar to the benefit of adjuvant chemotherapy. So if this is causally linked, this is something we cannot ignore, and it may have just as great an importance as our adjuvant chemotherapy. So where do we go from here? Is there evidence that physical activity is going to affect the adverse pro-inflammatory effects of obesity? The answer is probably a resounding yes. So a study of physical activity and diet compared to diet alone or physical activity alone showed that the two together led to the greatest weight loss, and also that there was a significant decrease in estrogens associated with weight loss. In breast cancer survivors, the SHAPE study of physical activity and diet showed that a weight loss of 5% significantly decreases both estradiol and estrone. So a number of biomarkers that correlate with estrogen and inflammation are significantly decreased in those individuals who are capable of losing weight through physical activity and diet. So we have reason to believe that there will be a causal link between weight loss, reduction of inflammation, and that might indeed correlate with better patient outcome. But it's not easy to get patients to lose weight. So there are a number of studies ongoing. I wanted to just briefly review them and end here. A number of things are ongoing, but I think I share the frustration of many practicing oncologists in that we see our patients come in, they're overweight to start with, they have trouble initiating or maintaining healthy behaviors during chemotherapy and treatment, their weight goes up, and then it's really hard to get the weight off and to lose weight. And I go over a diet history and weight in all my patients every time I see them. And I'm still finding that it's not that common for patients to actually change lifestyle. So lifestyle interventions are something I'm very interested in doing. One of the other compelling reasons for these lifestyle interventions is comes from some of our further studies in the lab. We've recently found in mouse models that high estrone in obese, diet-induced obese mice increases metastasis from breast cancer. We've tested that directly. We've also tested the effects of estrone and obesity on anti-tumor immunity. We find that estrone upregulates T regulatory cells, myeloid-derived suppressor cells, reduces effective numbers of CD4 and CD8 T cells, and also leads to upregulation of PD-L1, that ligand that bamboozles the immune system and is the target of checkpoint inhibitors. And indeed, we have some evidence from mouse models that that obesity can convert normally an immunologically cold ER-positive breast cancer model to a hot model. And we've found evidence in vitro that checkpoint inhibitors collaborate with anti-estrogens. So I know there are trials ongoing of this, but I think we have an opportunity here to initiate a new trial in unique populations and potentially with novel inhibitors. I'm actually currently studying together with an endocrinologist, Ronald Goldenberg, from Miami, as well as colleagues here at Georgetown. We want to see whether or not our hypotheses coming from the lab are true in women over time. And we have access to serial sera samples, over 10,000 in total, from 2,000 women who are overweight and had impaired glucose tolerance. And this study was initiated in the middle 90s, in the late 1990s. And we have serum samples that we're going to test estrone, inflammatory cytokines, and look at the lipidome and see how these correlate with development of breast cancer in this population over time. And this would provide compelling data that, that these relationships hold true in women. Chiranjit Dash is the PI and with myself and Lucille Adams-Campbell on a grant that we're just submitting to test relationships between estrone, cytokines, and oxidative stress indicators to see if they decrease in women accrued into the FIERCE study, which was a study of obese postmenopausal Black women who were either in an exercise or no exercise arm. And we're going to test whether are these relationships seen between estrone and inflammation, and are they in those individuals in which the intervention led to weight loss, does the estrone and the cytokine levels do they go down? So that's going to be a very interesting ongoing study. It's actually one of the reasons I will confess that I moved to Georgetown, is that I really saw a marvelous opportunity to interact with Lucille and Chiranjit Dash in these studies. And I'm just delighted to have them as collaborators. One of the ongoing pieces of work I mentioned in my lab is to look at how does estrone change how the estrogen receptor is recruited to chromatin and the transcriptome or profile of genes that are expressed particularly in the area of inflammatory mediators. This work has probably got relevance to liver disease, inflammatory bowel disease, cardiac disease, many endocrine disorders, as well as cancer. And I really look forward to collaborating with people here in the Department of Medicine on these kind of studies. And finally, lifestyle interventions. We've got to be able to figure out better ways of doing this. One of the other areas I'll mention in passing is the FXR receptor ligand that Moshe Levy has been working with has shown that FXR agonism attenuates inflammation in association with obesity. And it would be really interesting possibly to bring in a lifestyle intervention in which you can also collaborate, have a targeted therapy such as an FXR agonist to try to accelerate the attenuation of inflammation, which may actually be propitious for weight loss, as well as reducing the adverse effects of it. So that's another collaboration that I didn't officially put up on my slide, but I'm just wanting and waiting to develop better lifestyle interventions that really work to address the problem of obesity in our breast cancer patients. So with that, I will thank, I need to mention Rehanna Qureshi was the first author on the cell metabolism paper, and she and Manuel worked, both postdocs in the lab, really worked very hard over a long period of time on this. Massimo looked at the levels of inflammatory mediators in primary breast tissue, and other people in the lab contributed to the molecular biology. This was a collaboration with Mark Littman. I have to acknowledge Mark's amazing intellectual flexibility in even being willing to consider that estradiol might not be the main driver, and estrone is, since Mark made that first observation, it was a seminal observation in the 70s that ER-positive breast cancers are stimulated by estrogen to grow, and we can interdict that with anti-estrogen therapies such as tamoxifen. I've established marvelous collaborations with other investigators here at Georgetown, and with that, I'll take questions.